the chat window for any questions. Well, thanks everybody for joining. My name is Jejo Koola, assistant professor here within Biomedical Informatics, as well as a practicing clinician within hospital medicine for those that I haven't had the pleasure to meet during the first 2 weeks of this seminar. But it's my distinct pleasure to talk about you with something that I'm really excited about. And it's really kind of a, where the tires meet the road situation, some pretty advanced modeling with some real world implications. And this has to do with medical device safety and how to monitor some of these medical devices in a more sophisticated manner. So I'm just going to get into it and try to describe what the problem is, first of all. Before I do that, I want to go ahead and show my acknowledgement slide first and foremost, because this project is under the auspices of two PIs, Dr. Michael Matheny from Vanderbilt University and Dr. Fred Resnick from the Leahy Clinic, who are kind enough to donate some of their slides. And also some of this work is by Dr. Sharon Davis and Dr. Henry Sismagenda, Sharon from Vanderbilt and Henry from Leahy, who also were kind enough to donate some slides. So thanks to them, and also thanks to the NHLBI, who is funding this research. So first question is, I know it's been like a year or so, but I hope everybody recognizes these menaces, these scooters that we had in San Diego. And something interesting to, I guess, depending on who you are, a trend that occurred not just in San Diego, but all across the nation. And what we're looking at here in this graph is the dramatic increase in scooter-related accidents in America. Now, why would this happen? Was there something wrong with these scooters that were manufactured in 2017, 2018, and 2019, where scooter manufacturers perhaps creating models or outputting something from their factory that were inherently defective? Or was there potentially something that was related to user error? Maybe something that has to do with people hanging out in San Diego and OB or PB, and maybe deciding to get on one of these scooters at 1 o'clock in the morning and then end up in the emergency room because of an accident. And as you can imagine, there's a lot of user factors and factors related to the environment that may affect the outcome of these adverse events, in which case could have been serious, particularly for these scooters, which included head trauma and various types of bodily trauma. Now, this is the situation with just, something fun, scooters. But there's other systems that we use that we don't think about quite as clearly in terms of how well they work. And this is a system that is just starting to get investigated. Now, one of those electric scooters is pretty easy to build for these companies. At the end of the day, it's a set of wheels, a platform, and a small motor. But implanted medical devices is an absolutely monstrous market, not only with the US, but globally. And it can be something as trivial as just a crown that you get at the dentist office, all the way to an artificial heart. And like a lot of changes that the FDA has implemented over the last 20 years or so, there's been increasing effort spent on actually monitoring the safety of these devices, much like the safety of medication in a process called post-market safety surveillance. What that means is that, yes, the FDA has approved the device for use or the medication for use. It has passed the appropriate clinical trials, but the safety of the device or medication needs to be continued to be monitored. But when that happens, and there are adverse outcomes. For example, those scooters, we started seeing a whole bunch more accidents from those scooters from 2017 to 2019. Does that automatically mean that the scooter company was doing something wrong, or was there potentially other factors at play? And in this situation, there has been a lack of sophistication about how to identify what's really going wrong when something goes wrong with a medical device. There has been, however, a mandated nationwide registry for any time anything gets implanted into a human. So there's a little serial number and a lot number. If you work on healthcare, you'll see people document this in notes and surgeries. If you've been a patient who's had anything inserted as a medical device, you might have received some sort of card or pamphlet describing exactly what you had and a serial number. All that information is being gathered nationally for the purposes of post-market surveillance. But the problem is, how are we going to be able to use this information to drive better healthcare? And that's really the crux of what we're going to be talking about today. Because the truth is, there's a lot of differences that happen with adverse outcomes related to medical devices over time. So for example, here we are in this sort of bottom table looking at a couple of real medical devices that were studied. Some of these are kind of simple, straightforward medical devices, and some of them are very complicated. For example, an artificial aortic heart valve. And what these studies have shown is looking at the adverse event rate from when the device initially came on the market compared to the adverse event rate in the final year of the study. And in the very last column of that table at the bottom, you can see what the difference was in the adverse event rate. And what they've shown is that basically the adverse events went down over time. Now, why might that happen? So there can be several issues at play here. One of them, although this research study does not necessarily address this question, but one of the issues at play here may be changes in who, as in the patient, is getting the medical device. As you can imagine, the chance for an adverse outcome for any medical intervention is at least partly dependent on the health status of the patient. Just sicker patients are more likely to have adverse outcomes. So if there's a change in the event rate related to a device, it could be because maybe, for example, doctors are only using that specific type of device in extremely sick patients because it's their last hope. And just over time, the event rate goes up in that patient population for that device, but it has nothing to do with the device itself. It was just a selection bias in terms of who was getting the device. Or conversely, maybe doctors start using the device in maybe less sick patients, maybe patients who are more stable or who have earlier disease, and maybe save newer devices that have come on the market for patients who are sicker or who have more advanced disease. And therefore, you might see a change in the event rate over time, again, due to patient selection. But in this situation, that's not what we're talking about. We are talking about the two other aspects that has been difficult to disentangle. And the reason is this concept of patient selection and the selection bias, as it has to do with medical interventions, has been fairly aggressively studied over the decades. And how to go about addressing these issues. So for example, one of the fundamental reasons that we use randomized clinical trials when we evaluate drugs or whatever medical intervention is to try to eliminate that bias that certain patients might get a drug and certain patients may get a placebo based upon characteristics of the patient. And that's why you randomize it, and both the patient and the person doing the research study ideally are blind to the intervention. And similarly, in retrospective or observational studies, you might have heard of statistical processes such as matching, which attempts to disentangle that selection bias. So that's why we're not talking about the issue about selection bias or bias introduced from patient selection. What we are talking about today is trying to disentangle the most likely what we call learning effect versus what is intrinsic to the device itself. And this is just a fancy way of saying, hey, the more you do something, the better you get at it. You know, the old adage, if you do something 10,000 times, you can become perfect at it. Well, hopefully someone doesn't need to become, install 10,000 artificial hearts before they become proficient at that procedure. But certainly your ability to do that procedure safely and your patient to have good outcomes is likely at least partly related to your overall skill level and to how much experience you have with this particular device. So here is sort of an illustrative example of what we're trying to talk about. So again, this is mostly semi-simulated data for a potential real device called the MINX vascular closure device. The x-axis is time. The y-axis is relative risk. Basically, the higher the number on the y-axis, the more adverse events happened during that time period for patients who are getting this device. And what do you see? You see, well, when this device first started out on the market in 2011, there was some elevated risk, approximately a relative risk of 2, suggesting that this device compared to some controlled device or some older device on the market may be twice as likely to cause adverse outcomes, but the confidence interval crossed 1. And in terms of relative risks and similar statistics, what that means is that, yes, the point estimate of 2 saying that something was twice as likely to happen, but that was not statistically significant. So it's difficult to interpret. And what you see in those first 4 quarters, so those first 4 lines, 2011, quarter 1, quarter 2, quarter 3, quarter 4, is this kind of steady risk. But that confidence interval is still quite large. It crosses that threshold of 1. So not technically statistically significant, but what to do about that? And then all of a sudden in 2012, we start noticing that those confidence intervals get narrower and narrower. And now there's concern that this device, compared to some other device, some older device, can potentially cause harm for patients. And is that really what's happening? And what we're trying to look at here ultimately, and what was done with this particular project, is try to show that some of the change in the risk for this device could be related to this concept of the learning effect that the individual practitioner had. And part of the reason that the confidence interval narrowed was because of the more homogeneity that happened with practitioners as they kept using that same medical device over the years. Now, that being said, there were still issues within this analysis compared to the control device. So how are we going to try to disentangle that? And that's the crux of this issue from a modeling perspective. Certainly, there's a non-modeling perspective of just looking at the situation. There's existing systems in place in healthcare organizations to look at any adverse event or outcome that may happen to patients, to try to identify them on a case-by-case basis. But that's different than being able to look at a situation nationally. And at the end of the day, for us people who are in informatics, what we love more than anything is being able to analyze data. So I'm going to fast forward over that and go back to this example. So as I said, what we saw here is something happening with this particular device that over time, the relative risk, the confidence intervals sort of narrowed, became statistically significant, and the relative risk became statistically significant. But our goal here actually is to try to say, all right, well, how much of this change was due to the actual device itself versus how much of this change may have been due to the fact that different providers were using this device and getting more used to it. So what these two separate sets of dot plots with whiskers try to show is that, number one, the original change in relative risk over time, which, as we already discussed, was kind of flat between 1.5 and 2, and then the same relative risk that was further adjusted for the learning effect from the provider. That is to say that, hey, you know, this device was on the market for over 2 years. People got better at using that device. Therefore, the adverse outcome went down. But what is important to note in this analysis, or at least this illustrative example, is that, hey, even after accommodating for the potential improvement in adverse outcomes, after that device has been on market for a few years, there was still excess adverse events that could not have been attributed to the operator or also to the patient. And is that a safety signal that should be reported to the FDA for further evaluation? So this project, I'm going to give you now the overview. And before that, I'm going to pause for 1 second. I realized that was a fairly extensive background, but it's really important to understand, you know, where this sort of situated within the bigger picture. So I'll pause for 2 minutes to see if there are any questions. Okay, all right, keep going. So this project starts out in 3 phases. And what I'm going to really be talking about today is really the first 2 phases. The first phase was a process to create an artificial test system. Because this kind of research for this situation has not been well investigated, what we wanted to be able to do is really understand what we're able to detect signal-wise in a perfectly artificial system where we control the process. And I wanted to talk about this a little bit because honestly, creating purely artificial data or synthetic data is not something that we often do. In fact, I personally, before being involved in this project, had not done that. Most of the time, when we're attempting to get data to create a model, we're usually looking for real-world data somewhere from somebody. But artificial data has some benefits, and we'll talk about that. And the second step of this project is to try to develop methods to actually disentangle what we talked about in the background. And the third step of this project, or aim 3, is to start doing this with real devices in real time, although I'm not going to be talking about aim 3 during today's presentation. So what is this aim 1 and the data generation process? I'm going to skip over a couple of these slides and go to something that's a little more graphical. So the whole point of aim 1 is to look at a system where we can start out with basic a priori information that we control and slowly layer on complexities, and at the end of the day, create a completely synthetic data set. So what are some of the things that we're going to control? Well, at the very bottom, kind of like a pyramid, is synthetic data about patients. The second level is to identify which patients get a certain device, device A versus device B, kind of like saying who gets the research drug versus a placebo. The third step is to estimate, or not estimate, but specify some baseline risk. Then we try to layer on some information about, and when we say operator, what we're really talking about is the doctor or individual healthcare provider who is implanting the medical device. The next level of complexity is some information about the institution. And finally, there's going to be just noise in any real world data set, so we can simulate that as well. So I'm going to go through some of this stuff. So what are some aspects about patient data generation when it's purely synthetic that is important for us to consider? Well, there's some obvious things, such as, is it modeled upon real world electronic health record or real world patient physiology, or is it completely synthetic data? And if it is synthetic data, how is that being generated? Also, taking into account various aspects of real world retrospective electronic healthcare data, such as missingness or just lack of relevant predictors or variables that may influence the outcome, which can cause you to have confounding. The second aspect in terms of device assignment is to try to figure out, again, who's going to get the device that is probably in a real world scenario, because again, we're trying to create synthetic data that is mimicking to some extent what happens in real world. And in the real world, the chance of getting device B versus device A, as I mentioned, if you're not part of a research study, is probably going to be a biased assignment, because the healthcare provider may choose, for example, the newer device in sicker patients or in less sick patients, which will bias your outcome. So you would want to try to control for that when you're generating the data. And then some of these other steps I'm going to skip because I think they're fairly standard, although I wanted to sort of quickly mention for this initial phase, the operator or healthcare provider characteristics. The main ones that we're focusing on is just the temporal aspect. In other words, how many times have you done this procedure overall? Like, for example, put in a stent to try to fix somebody's heart attack. And number two, how many times have you used this specific device? So let's say manufacturer A makes this kind of stent, manufacturer B makes a different kind of stent. How many times have you used the stent from manufacturer B? And similarly, you can imagine the kinds of things about institutional characteristics, such as UC San Diego versus the University of Colorado versus maybe a community health hospital or a non-academic hospital, and how some of those characteristics may affect the adverse event rate as it has to do with procedures in general and this specific type of device in particular. So there are, I should note, lots of published data sources available for some of this device data. Again, a lot because of these national registries that have been designed. And eventually we want to be able to use that data. But before that, what we really want to do is to try to simplify the problem. So one level below using data for the real device would be just using what we have in our electronic health records, because we have direct access to that, we can manipulate that. So one aspect of this project is going to be focusing on using our electronic health record data to generate simulated patient data. But again, we can control what the actual adverse event rate is when we generate synthetic data based upon real world electronic health record data. And there are different sources to that. I'm actually not going to go into it. I think at some point during your informatics career, some point during your career, you're going to hear about these electronic health record data sources. So I'm not going to belabor that point. But I do want to just talk about this data generation process briefly, because again, the thought of generating synthetic data seems trivial at first, because you're thinking, okay, well, I'm just going to use an Excel file. I'm going to use the random number function in Excel. I can do that for a few times. Boom, got some synthetic data. This should be easy. Well, the issue is that what you want to do is not just generate synthetic data, but synthetic data that has defined relationships. Because our goal here is to be able to have two teams. One team that's kind of the bad guys that are generating the synthetic data. And then there's team B that's kind of playing Clue, that's kind of the detectives that are getting the synthetic data, but they don't know what parameters were used to generate the synthetic data. And it's their job to figure out, is there or is there not this learning effect? Or is there excess adverse event attributable to a certain device, device B, compared to device A? And if so, Is it something to do with the device or potentially something to do with the operator? So how does that happen? So how are we defining this? Well, this is, again, sort of a bottom-up process. So the very bottom layer is going to be the patient-relevant data, the procedure-relevant data, and provider-relevant data. So those are the variables that we control. So during the synthetic data generation process, we are in this situation. Remember, at this point, we're not even trying to mimic electronic health record data. This is pure synthetic data. So for example, we have 3 variables that are supposed to represent, something about a patient. In this case, one of the variables is normally distributed, one is binomially distributed, one is Poisson distributed. We have a set of variables that has to do with the procedure itself. And again, at this phase, the most important aspect of that being just a temporal order of how many times a specific individual has actually performed that procedure with that device. And then finally, the purple square is just something about the provider or the hospital setting. And again, all of this is synthetic. We're just making this up. The next step is actually combining these into some sort of probability. So for this one, the first step is to create basically a logistic model based upon all those different patient variables. So that normal variable, that binomially distributed variable, and Poisson variable. What I'm showing you in this combined probability layer are the odds ratios that we pre-specify when we create synthetic data for these variables, which again, we've pre-specified where the outcome is an adverse event. So what this is saying is that device B in this pretend data set is twice as likely to cause an adverse event compared to device A. And similarly, it's saying something about the normal and Poisson and binomial distributed variables. And then finally, still within that combined probability, that second equation you see P1 equals P0, what we're basically doing there is trying to model that learning effect, where P0 is the probability of an adverse outcome based upon that pre-specified logistic equation with those odds ratios you see on the screen. And then there is potentially increased adverse event rate depending on if we're trying to simulate a learning effect or not. And that's what that term B0 times T to the negative B1 is. So all that's saying, all that sort of complicated way of saying is there's some baseline chance of something going wrong based upon how sick the patient is, plus some chance of something going wrong based upon how many times the doctor has done this procedure with this particular kind of device. And then finally, when we generate the data, the very last layer at the top, our outcome, is what we call a Bernoulli distribution, where we're basically drawing zeros or ones with the probability of P1. So it's basically we're saying we're flipping a coin, but unlike a coin where theoretically heads or tails is 50-50, this coin that we're flipping for every single patient, every single procedure is based upon that final probability from the middle layer. And when at the end of the day, this is all that looks like. This is literally some of the synthetic data that we're generating. And all it is a bunch of made up numbers combined into pre-specified relationships like we talked about. So what does this end up looking like? So let me walk you through this graph, because at first glance, it's a little bit challenging. So the x-axis here is the procedure number. So this is the number of times that an individual has used that particular device to perform whatever procedure, right? The device itself, there's 2 of them. There's device A and device B. And again, this is all made up data. There's device A and device B. Device A are the circles. Device B are the squares. And finally, we are looking at two situations. One situation when we a priori specify a learning effect, and one situation where we say there is no learning effect. So that last part is going to get kind of confusing. So let me describe what I mean. Let us look, and oh, by the way, the y-axis is the probability of something bad happening, our outcome probability. Let's look at those two bottom lines, or those two bottom ribbons. You'll see that it's all circles, but one ribbon is this sort of aquamarine color, and one ribbon is this pinkish salmon color. Well, what do the circles mean? The circles mean device A. And what do the two colors mean? Well, the circles that are aquamarine are the outcome, basically the probability of something going bad if there was a learning effect, if we are pre-specifying a learning effect. And then the pink circles is basically the outcome if we are not pre-specifying a learning effect. But in this synthetic data set, device A is the device that we're pretending has been on the market for 20 years. And in this situation, device A, again, we are synthetically generating this data. In this situation, device A does not have a learning effect built into it. We're assuming because this thing's been on the market for 20 years, people are super used to it. They know what they're doing, no big deal. The chance of adverse outcomes is no different whether it's like your first time using it versus your 200th time using it. Now, that's different for device B. In this made-up example, the synthetic data, device B is the one that we're pretending is the one that's brand new. That's the one that's like the fancy stuff. It just came on market. So for that, let's look at the two top ribbons. So again, those two top ribbons are squares. So that means it's device B. There's a salmon one and there's an aquamarine one. And again, the salmon means a priori, we're specifying no learning effect. So if you look at the squares and the salmon, what we see is, give or take, given the fluctuations due to noise, it's relatively flat. And again, we've designed it to be that way. Whereas the one that is aquamarine, there is just a very slight hint of decreasing adverse events compared to like the very first time somebody uses that device to like the 200th time somebody uses that device. You can ignore the middle two ribbons. The middle two ribbons are just looking at what happens regardless of whether it's device A or device B, but it's less illustrative. At the end of the day, what we're trying to figure out is which scenario is it. Remember, there's like the team that's creating the data, and then there's like team B, those are the detectives that are blinded to what's happening behind the scenes and trying to figure out, okay, is this scenario aquamarine? That is to say, there's a learning effect, that is the operator or the doctor is going to get better over time, or is this scenario with this data salmon? That means there is no learning effect. Like whatever adverse outcomes there are due to the device and due to the patient themselves. So I'll pause right there again just to see if there's any questions before we go into some of the actual modeling and results.
I have a quick question. Can you hear me?
Yes,
okay?
I can.
So on the top two ribbons, why is there such a big jump between the second to last point and the top point for the aquamarine color?
Oh, great question. That's just an artifact because that last time point did not have as many observations. So this graph is generated using simple binning. So in other words, what that means is it looked at all the devices that were, all the procedures that were done between like day 0 and day 25 and day 50, blah, blah, blah. And then it looked at the average outcomes. So let's say, 20 heart stents were placed those first 25 days, and 5 patients had an adverse outcome. That means the adverse outcome event rate for that first bin would be 25%. But the very last bin in the synthetic data just had a lower count. Therefore, the point estimate has more variability and the confidence interval has more variability. Does that make sense?
Yep, thank you.
No, thank you for the question. It's a really great point. Yeah. Great, I'll go ahead. So the first thing I'm basically going to talk about is the simplest model, which is a regression model, and more importantly, what we call a parametric model. So when I started out this talk, the title was using parametric versus non-parametric methods to assess learning effects. And lo and behold, it's minute 43, and I haven't even told you why that was important. So the first step is from a parametric versus non-parametric standpoint. The first step that we're going to try to do is try to model this parametrically. So I'm going to ask the question, has anyone ever used that word parametric versus non-parametric? And what does that mean to you? And anyone can speak up in terms of any kind of data modeling, and why is that relevant? What would be maybe the pros and cons of those two approaches? Well, I can give it a multiple choice. One of the issues may be ease of use. A second issue may be handling nonlinearity. A third issue may be complexity of the data. And a fourth issue might have to do with fidelity with the underlying true gold standard. So the first step in this modeling process, or using a parametric approach, is ultimately trying to say, can we understand what is happening behind the scenes explicitly? That is to say, can we create a mathematical equation that explicitly says that the chance of an adverse outcome is related to these variables in this specific way? And the way we've done it for this situation is using a simple regression model. You can do it more complicated ways, but this is the most common form of doing sort of parametric modeling. This is what's called a general linear model. I'm going to go briefly into it just to talk about how we approach this from a parametric perspective. So there's basically some baseline data. What we're looking for in that top table is the cohort. We see that about 60% of people got device A, about 40% got device B, blah, blah, blah. Then the bottom table tries to look at the event rate, and so many patients had a bad outcome with device A, so many patients had a bad outcome with device B. Straightforward. In this particular data set, for example, again, this is all synthetic data. There's 25 institutions or hospitals that were pre-specified. Each institution or hospital has 15 doctors doing procedures, and there's some variability in how many procedures each doctor has done. And those were, again, all pre-specified based upon sort of standard parameters. We said, we're going to randomly choose the number of procedures somewhere between 200 and 250 or whatever it is. So the first step in any parametric modeling is, again, handling this issue with selection bias. I'm not going to go into this process, only to say, just to allay concerns about methodology. The first step whenever you have retrospective data, and particularly looking at two interventions, and even though this is synthetic data, this is still what we would call retrospective data. If you have two interventions, as we discussed, it can be confounded, the probability of an adverse event, something going wrong with one intervention, device B, over another intervention, device A, it may be confounded by something other than a device. Again, the main one being selection bias by the patients. And the way that's generally handled is through a process called matching. I'm not going to go about how this was done for this particular data set, but just FYI, we went ahead and did matching. What I'd like to be able to talk about is, now, given that we have this data set now, how are we going to try to find out if there's a learning effect using a parametric approach? So we built a few different models. So the first model is a logistic regression model with the outcome just being 0 or 1. Did this patient getting a procedure, this procedure, have a bad event or not? Where the predictor variables were the 3 synthetic patient variables. So again, these were completely made up, but let's say, for example, the patient variables were A, a history of diabetes, B, the patient's body mass index, and C, what was their blood pressure on the morning of them getting their surgery. And then you try to solve for it. And then what this result is just showing is that in this particular synthetic data set, ignoring anything about the operator or the institution or whatever, just using those variables that have to do with the patient, variables 2 and 3, again, they're synthetic, were statistically significant. That's what the table shows. Synthetic variable 1 was not related to the outcome. And remember, the team that is analyzing the synthetic data is separate from the team that's generating it. So the team that's analyzing it does not know what the team that generated it did. They don't know if they said there was a statistically significant relationship between variable 1 and the outcome or not. So that's scenario A, that's only using patient-specific factors. Scenario B is using patient-specific factors as well as operator volume. All that is saying is, again, like looking at that data for Bob, who's getting surgery on August 10th, by Dr. Wendy, how many times has Wendy done this procedure? And simply using that as a numeric indicator, like this is the 123rd time Wendy has done this procedure. And again, creating a logistic regression model. And what this suggests in this particular analysis was that it's the same thing for the patient-specific factors. F1 was not, or variable 1 was not statistically significant. Variables 2 and 3, the patient-specific ones, were statistically significant. And finally, the operator case number, in other words, whether the doctor had done this 1 time only or 200 times, was also statistically significant for device B. And then the estimate of the regression model is negative for that case number. So what it means is that for every additional time that a doctor does a procedure with this device, the chance of an adverse outcome goes down. And then finally, device or scenario C is very similar to scenario B. The difference is it's not just how many times has that individual doctor done the procedure, but also how many times has that hospital used this device or done that procedure just in general. Again, the idea being that increased experience, not only for the provider level, but a systems level, is likely to affect adverse outcomes. And what we found in this sort of very simple analysis for this sort of trying to do a detective story, right? Because again, the team that's analyzing the data does not know how the data was generated. And in this case, for each of those 3 scenarios, which is what we see in this table, the detective team looked at a few different ways of evaluating the model, and then finally said that model B is the best model, where model B being the model that had the patient-specific variables, like I said, we're just going to pretend it's going to be like blood pressure and weight and history of diabetes, plus the operator-specific volume, like how many times have they done that procedure up to that point. Whereas scenario A and C were potentially inferior. So there was one attempt at trying to identify, kind of a rudimentary attempt, but an attempt to identify that learning effect was present. Of note, this does not try to say what does that learning effect look like. It just says that there was a learning effect. Okay, so that was a ton of talking, but what we ended up talking about is this entire process. We generated, a bunch of synthetic data. We divided it up into device B versus device A. We did matching. We created a model, et cetera, et cetera. We did this parametric modeling with logistic regression, and the system said, yep, there's a learning effect. We should use this to do something, drive policy. Maybe we would say, okay, doctors need to be supervised until they do at least 20 procedures with this device, because our study shows that once you get to procedure number 20, like the learning effect stabilizes and plateaus or whatever. But what happens if this parametric modeling does not find a learning effect, right? Let's say like none of these variables were statistically significant for operator volume. And that's where the non-parametric assessment come in. And again, the question was, why would we use non-parametric models in these situations? Well, there are a few different reasons. The number one reason, again, is that some of the parametric models, by definition, there's an old adage in statistics, anytime you make a correct assumption about your data, you're always going to get a better model, right? But that's the problem. Most of the times we don't make correct assumptions about our data. We often violate basic assumptions about our models to some degree, because particularly with EHR or health data, there's just too many variables. I mean, it's human physiology we're trying to model here. So it's challenging to do that with parametric models. When you make a parametric model, you have to make correct decisions and assumptions about the underlying data. And that can be the power of non-parametric, or what you may choose to call machine learning. It doesn't necessarily have to be a quote unquote machine learning model, but a model that is non-parametric. In other words, it does not require you to make as much assumptions about the data. And therefore, if you do not find a learning effect with the parametric model, it may be because there is no learning effect, but it may also be because you made incorrect assumptions when you built that parametric model. So then our goal was to incorporate a non-parametric model. And the other issue is that the non-parametric model, at least within this framework, we wanted to be able to use some of the underlying time varying nature of this data. Like all healthcare data almost always ineluctably has some sort of time varying component, because we're getting repeated measures on the same patients, right? Bob comes to clinic on August 1st, he goes to surgery on September 15th, et cetera. And sometimes there are non-parametric or machine learning models that allow us to inherently model this time varying nature. And again, I already talked about some of these nonlinear effects. So I'm running out of time here, so I'm briefly just going to talk about some of the early attempts that we've looked at for these non-parametric models. So I already talked about regression. We just spent 15 minutes looking at that. And regression, remember, in this context, is a parametric model that we created. We also looked at just a random forest model. For those that haven't been exposed to some of these models, random forest is a kind of a bread and butter machine learning model, particularly when you're interested in modeling nonlinear interactions, and it's very robust to various violations of assumptions. The second machine learning model was a multi-layer perceptron, which is kind of a really simple neural network model. And then finally was an LSTM, or long short-term memory neural network. And the only thing about that is a model that is what we might call temporally aware. The way that the model is constructed actually tries to incorporate something about the dynamic nature of what is happening with a patient's health status. So I'm running out of time, so I'm going to skip some of these slides. And what I'm essentially going to have to sort of fast forward to is at least early results. So far, we have not been able to identify for these particular early data sets a benefit of the machine learning models over the parametric model. So what we've tried to do is to, change various parameters and then synthesize these data sets. And at least within these data sets, the parametric model does just as well as the machine learning model. Now, that's not necessarily a bad thing. We want the parametric model to work well, but what we are ultimately interested in is that failure state. Like, what are the situations where the parametric model is not able to find a learning effect, but the machine learning model is able to find a learning effect, and how do we handle that? So that is the eventual goal of AIM-2, to try to figure out that process. And once we're able to do that, our eventual goal again is to use those knowledge to go on to AIM-3, which is actual real-world prospective surveillance of new medical devices as they come onto market, to try to identify adverse events and potentially identify adverse events that have to do with the device itself, which might trigger a safety report, or adverse events that might have to do with operator proficiency, which might help provide guidance for how a healthcare organization and or individual doctors go about practicing their healthcare. I'm going to go ahead and stop there. I realize I'm just on the edge of time, but I'm happy to stay a little late and answer any questions if anybody has them.
Jejo, you